we continue our glycolysis. We started off yesterday where we considered the different metabolic processes that actually go on in the body, and if we look at the first slide here, the overall catabolic processes look at the breakdown of lipids, polysaccharides and proteins. Now, the breakdown of lipids gets into components of fatty acids and glycerol. The polysaccharides break down to monosaccharides and the proteins break down to amino acids. Now, in the anabolic processes, we have these broken down amino acids and other factors that actually get on into building up the other macromolecules that are required for our bodily functions. Now, what we are interested in is the breakdown of the monosaccharides, particularly the process of glycolysis that takes glucose and breaks it down into pyruvate, and later on we will see how this pyruvate then gets on into the tricarboxylic acid cycle or the Krebs cycle to finally get to water and carbon dioxide. And an offshoot of that is the production of ATP, where we studied oxidative phosphorylation in the different complex processes that require a number of electron transfer cofactors as well as certain enzymes. The overall equation for glycolysis is the breakdown of glucose into two pyruvates. Now, what we have here is we see how ATP is produced. We will see as we continue with all the steps, we will see how ATP is produced in some of the steps, but in the first set that we did, we found ATP consumption. This is the number of steps that we considered in our last class. We had glucose going to glucose 6-phosphate, the enzyme being hexokinase. We have to remember that a kinase is a transferase that transfers a phosphate group. So, in this process we had the breakdown of ATP to ADP and the phosphate was transferred to the glucose. This then went on to form the ketose from aldose. So, we had fructose 6-phosphate that had the enzyme phosphoglucose isomerase acting on it, because this is an isomer, the aldose and the ketose that we have here. The fructose 6-phosphate then went on to form fructose 1,6-bisphosphate, where we required another ATP to be broken down, and the enzyme used there was another kinase, but in this time it was phosphofructokinase. After this particular step, we have aldolase come into the picture. Aldolase is what actually breaks up the 6-carbon membered ring into 2 3-carbon membered rings, where we have glyceraldehyde-3-phosphate and dihydroxyacetone phosphate. Now, these two are intertransferable by the enzyme triose phosphate isomerase, and we also learned that the equilibrium of this enzyme is such that dihydroxyacetone phosphate is preferred to be formed in larger quantities, but to shift the equilibrium to the right, where glyceraldehyde-3-phosphate will be utilized, because that is what continues the glycolysis process. So, we have to have glyceraldehyde-3-phosphate that is going to continue the glycolysis or continue with the breakdown, and we have to remember here that we now have 2 3-carbon units instead of the 6-carbon unit that we started off with. Okay. So, this is our step number 6. If you remember, we reached glyceraldehyde-3-phosphate after the action of aldolase on fructose 1,6-bisphosphate. So, our next step is the formation of 1,3-bisphosphoglycerate from glyceraldehyde-3-phosphate. So, we are now adding a phosphate, but we have not used ATP in this step. We are using PI. Okay. So, the reaction here is glyceraldehyde-3-phosphate plus NAD plus PI in a reversible step, giving you 1,3-bisphosphoglycerate plus NADH plus H plus. So, this is our 6th step, where what has been done is we have added a phosphate using NAD plus forming NADH in a reversible step. Now, in this case what happens, this is the only step in glycolysis where NAD plus is actually reduced to NADH. In this step, that uses Glyceraldehyde-3-phosphate dehydrogenase, because you have this hydrogen being removed from the aldehyde group and you have a phosphate introduced by the Pi that comes in as a factor to form 1,3-bis phosphoglycerate. And what we have here now is, if you notice, we are going to actually create an acid from this, okay, because this now has the form where we have C double bond O PO3 2 minus. Okay, this was initially an aldehyde, okay, and we are gradually getting to an acid. Now what is going to happen is, in this case there is a cysteine thiol. All of us know that in the cysteine residues we have the SH thiol group. Okay, now what happens is, the cysteine thiol is present at the active site of the glyceraldehyde-3-phosphate dehydrogenase, and that actually forms an intermediate with the acetaldehyde of the glyceraldehyde-3-phosphate, the aldehyde, that reacts with the cysteine thiol to form what is called a thiohemiacetal. Okay, so what happens here, like we learnt yesterday in one of the reactions, we had lysine come into the picture. Here we have cysteine. We will also see one of the enzymes has histidine. Okay, so these are the particular side chains of the enzymes that are going to take part in the overall reactions for the transformations to occur. Now in this case what we have is, we start off with, let's get this up here. Okay, so what we have is, here is our enzyme with the cysteine group attached to it. It's not working. Anyway, what we have here, you can see the enzyme has the SH group attached to this. Now what is happening here, this SH creates a thiohemiacetal intermediate by reacting with the aldehyde of the glyceraldehyde-3-phosphate. Okay, so this is our glyceraldehyde. So this is carbon number 1, carbon number 2, carbon number 3. Okay, because now remember, we have broken down our glucose into a 3-carbon system. So the enzyme, the cysteine group that is present in the active site of glyceraldehyde-3-phosphate dehydrogenase is forming a thiohemiacetal intermediate with the aldehyde of glyceraldehyde-3-phosphate. Okay, so now we have this linked to the enzyme. Okay, so now this part is linked to the enzyme. Then NAD plus is reduced to NADH. In the process we get an acyl thioester intermediate. Where is this thioester? You see the CHOH is now C double bond O, and the S is part of the cysteine, so it becomes a thioester, an acyl thioester intermediate. Okay, now what happens is, then the Pi, that is the phosphate, comes and cleaves the SC bond, and your enzyme is regenerated. Okay, and in the event what happens is, the phosphate adds on to the first carbon atom. So now you have 1,3-bis phosphoglycerate. Is that clear? So what happens is, we have this acyl thioester that is attacked by the Pi, which actually cleaves the substrate, or rather in this case it has now become the product, and regenerates the enzyme. So we have again enzyme cysteine with SH that now is ready to act upon another glyceraldehyde-3-phosphate. Okay, so basically what we have here is, from glyceraldehyde-3-phosphate, with the help of this cysteine of the enzyme, we have the formation of a thiohemiacetal intermediate that then forms an acyl thioester intermediate, which then is attacked by Pi that yields 1,3-bis phosphoglycerate and regenerates the enzyme. Okay, So that is our step. So now we have landed up with 1,3-bis-phosphoglycerate. Now what happens is this phosphate is taken up by ADP to produce ATP. Okay? So we have now the first reaction where we have produced ATP in our glycolysis step. Okay? So ADP, one of the phosphates from 1,3-bis-phosphoglycerate, is the first one is lost to ADP, which produces ATP. In the event it forms 3-phosphoglycerate. So we now have an acid here, not an aldehyde, at carbon number 1. Okay? And all of this, remember, is enzymatic. Okay? So initially we had, what was the first thing that we had here? We had glyceraldehyde-3-phosphate. So this was our aldehyde. I have to create pyruvic acid. Okay? So I have to create an acidic group at position number 1. The first step in doing that is the formation of 1,3-bis-phosphoglycerate. In the formation of 1,3-bis-phosphoglycerate, the enzyme glyceraldehyde-3-phosphate dehydrogenase utilizes a cysteine thiol to bring about the reaction. And in the next step, we lose the phosphate to ADP, which produces ATP, and we have 1,3-bis-phosphoglycerate plus ADP go to 3-phosphoglycerate plus ATP. The next step is a mutase. If you remember, we considered in our last class all the enzymes that are actually going to take part in the glycolysis steps. The mutase shifts the phosphate from one position to the other. So the mutase enzyme shifts a moiety from one carbon atom to another. In this case, the 3-phospho is shifted to the second carbon. So the product is 2-phosphoglycerate. Okay? So instead of 3-phosphoglycerate, after the action of this enzyme phosphoglycerate mutase, which shifts the phosphate moiety from the third carbon atom to the second carbon atom. So the phosphate is shifted from the OH on C3 to the OH on C2. So we have a shift here. Now in this case, from 3-phosphoglycerate to 2-phosphoglycerate, the enzyme, which is phosphoglycerate mutase, has a histidine. Okay? And this histidine is present again where? In the active site of the enzyme. And what it does is, it helps in the phosphate transfer by accepting the phosphate and then donating it again. Okay? So the mechanism is such that the histidine accepts the phosphate from 3-phosphoglycerate and then donates it back, but to the second carbon atom. So this would obviously depend upon the position of where the carbon atoms are actually located in the active site of phosphoglycerate mutase. Okay? And in the intermediate that we have here, we have an additional actually 2,3-bis-phosphoglycerate, and this finally loses the phosphate, that's the third carbon atom, into becoming 2-phosphoglycerate. So we've already formed the acid. We're gradually getting to the step where we are going to form an enol and then a pyruvate. Okay. Now in the 9th step, we have enolase. Enolase, actually we have now reached 2-phosphoglycerate. We had 3-phosphoglycerate, we have now formed 2-phosphoglycerate by what enzyme? A mutase. After 2-phosphoglycerate, we have now an enolate intermediate. What is that enolate intermediate? It has formed, with the loss of H plus here, it has formed a double bond at this position between carbon atoms 1 and 2, we have a double bond formation. Then with the loss of OH, we have a double bond between carbons 2 and 3. Okay. So this negative charge of the oxygen will come back here, this forms a double bond here, and the OH is lost. Is that clear, the mechanism? So we have the O come back here to form the double bond, then this double bond shifts here, the OH is lost. So what we get is we get phosphoenol pyruvate. So we are gradually getting to our final step, which is going to be the formation of pyruvate from where? From glucose. Okay. So we have in our 9th step enolase that catalyzes the formation of phosphoenol pyruvate from 2-phosphoglycerate, which is actually nothing but a dehydration reaction. So essentially you are losing this hydrogen and this OH, okay, into forming a double bond between carbon atoms 2 and 3. That is essentially what is happening. So you have CH2 double bond C with the phosphate attached and CO minus, a carboxylate group. Okay, so essentially you are having a dehydration where you are losing this hydrogen and this OH, and you are forming a double bond. Now what else do you have to lose to form the pyruvate? The phosphate. So that will be our next step. But there are some other notes here where we have the dehydration reaction is magnesium dependent. The two magnesium ions interact with oxygen atoms of the substrate carboxyl group at the active site. So what happens is we have a magnesium interacting with the oxygen atoms of the carboxylic group of 2-phosphoglycerate, that helps stabilize. So we have 2 O minuses here, right? So what happens is the magnesium ions stabilize this enolate ion, and then we have an extraction of the hydrogen from carbon number 2 when a lysine actually helps in doing that. Okay, so the active site in this case would be the presence of the lysine and the presence of 2 magnesium ions that would help stabilize the enolate intermediate. So our next step now is the formation of pyruvate, where we have to lose the phosphate. Okay, so what we have is, this is the last step where we have formed pyruvate. Pyruvate is CH3 C double bond O COH, that is pyruvic acid. So we have phosphoenol pyruvate that was formed from what? It was formed from 2-phosphoglycerate. From 2-phosphoglycerate we formed phosphoenol pyruvate, and we now know that to form pyruvic acid we have to have CH3 C double bond O COOH, or rather pyruvate would be CH3 C double bond O COO minus. Okay, so we have to lose this phosphate, and who takes up the phosphate? ADP. Okay, so now we form another ATP here in this step of glycolysis. So, and the enzyme here is pyruvate kinase, that is shifting the phosphate. Remember, the kinase shifts the phosphate. It is taking the phosphate from phosphoenol pyruvate and giving it to ADP and forming ATP. The intermediate is enol pyruvate that actually then forms the pyruvate. So our last step is phosphoenol pyruvate plus ADP going to pyruvate plus ATP. Okay, so eventually we've broken down the glucose and we have finally formed pyruvate. This pyruvate is then later on going into, through acetyl CoA, it is going to go into the tricarboxylic acid cycle, and from there we will have the production of carbon dioxide and water, which we will see in a later class. Okay. Now the formation or the phosphate transfer from PEP, what is PEP? Phosphoenol pyruvate, okay, to ADP is a spontaneous reaction. Okay. PEP has a larger delta G of phosphate hydrolysis than ATP. What does that mean? It is going to lose the phosphate very easily, and ADP will take up this phosphate to form ATP, and because you have these anionic residues, there are certain cations that bind to the anionic residues at the active site of pyruvate kinase to bring about this particular reaction. Okay? So, remember that apart from the electron transfer cofactors, apart from the side chains that are present, since we have carboxylate anions or enolate ions, we have some cations specifically that are used to stabilize the intermediates. Okay? So, if we look at the overall glycolysis steps now, we have 10 steps. Okay? So, if we just reiterate, we have glucose in the first step going to glucose 6-phosphate. You have to remember that you have to form the pyruvate from the glucose. So, the first step is glucose with the help of hexokinase and ATP forming glucose 6-phosphate. The next step is an isomerase where we form fructose 6-phosphate. We then need to form fructose 1,6-bisphosphate, which means that we need another kinase and another ATP. Okay? This you should remember in this form. So, we have glucose 6-phosphate, and since you have to form fructose 1,6-bisphosphate, you have to form fructose 6-phosphate. So, you need an isomerase that is going to form fructose 6-phosphate from glucose 6-phosphate. With the kinase, you have fructose 1,6-bisphosphate, and aldolase acts upon the open ketone form of fructose 1,6-bisphosphate. That is what I showed you yesterday with the lysine acting there, and we have now a breakup into two 3-carbon moieties. They are dihydroxyacetone phosphate and glyceraldehyde-3-phosphate. The glyceraldehyde-3-phosphate is going to continue the glycolysis. So, what is our glycolysis continued? We have glyceraldehyde-3-phosphate with glyceraldehyde-3-phosphate dehydrogenase with the help of NAD plus and PI form 1,3-bisphosphoglycerate. Okay? So, you remember in this step when we have the dehydrogenase, you are forming from the glyceraldehyde, you are forming glycerate. That is important because you have to form finally pyruvate, which means you have to have what moiety? A carboxylic acid moiety, right? So, this is the step where you have that oxidation actually take place from the aldehyde to the acid. Okay? So, if you have a redox reaction, what is the enzyme that you need? A dehydrogenase or an oxidase or some such enzyme that is going to bring about the redox reaction, and for that you need an electron transfer cofactor, which in this case is NAD+. Okay? So, this is what you have to remember, where you have glyceraldehyde-3-phosphate that takes up NAD+ and a PI to form 1,3-bisphosphoglycerate. Okay? Then it has to lose the phosphate, as simple as that. Okay? So, now we needed the bisphosphoglycerate to oxidize the aldehyde. Okay? So, for the oxidation of the aldehyde, which in turn reduced NAD+ to NADH, okay, because it is a redox reaction, right? What is getting oxidized in this case? Glyceraldehyde is getting oxidized to form glycerate. What is getting reduced? NAD+ is getting reduced to NADH, and the enzyme is the dehydrogenase. Okay? So, we have glyceraldehyde-3-phosphate going to glyceraldehyde-3-phosphate dehydrogenase, and in the event it forms 1,3-bisphosphoglycerate. Now that you have formed the glycerate, you have to lose the phosphate to form the pyruvate. Okay? So, the first step in the loss of one of the phosphates is a phosphoglycerate kinase that is going to lose the phosphate that is attached to the carboxylic first carbon atom of the glycerate. Okay? And you have 3-phosphoglycerate formed. Fine? After you form 3-phosphoglycerate, there is a mutase reaction which shifts the phosphate moiety from the third carbon to the second carbon. So, you have 3-phosphoglycerate form 2-phosphoglycerate, then another enzyme that helps in the dehydration is enolase that results in phosphoenol pyruvate. So after phosphoenol pyruvate, you have the enolic form of pyruvic acid, which then forms pyruvate after the loss of the phosphate. And who takes up this phosphate? ADP. Okay, so that comprises the whole series of steps where glucose is broken down into pyruvate. Okay, now what we have to see is we have to do a balance of energy. Okay, we have to see how many ATPs are taken up, how many ATPs are produced, and to see whether the actual breakdown of glucose is giving us any energy at all. Okay, there is one thing that we have to remember, that per glucose there are 2 glyceraldehyde-3-phosphates. Okay, because what is the previous step? We have 2 of these. Okay, and in the triose phosphate isomerase, we know that the equilibrium is shifted to this side because this is being consumed in the further steps. Okay, so our balance sheet for the phosphate bonds of ATP. How many ATP bonds are broken? How many are broken? This is our whole series. We have one broken when we form glucose to glucose 6 phosphate, right? Another broken when we form fructose 1,6 bisphosphate from fructose 6 phosphate. Fine. So our answer for the first question is 2. How many phosphate bonds of ATP are produced? How many are produced? Is it 2 from every molecule of glucose? 4. Why? Because we have 2 of these. Glyceraldehyde, 1 glyceraldehyde-3-phosphate is going to create 2 ATPs, right? But in our previous step, what do we have? We have 2 of these moieties because we have broken down what? The 6-membered ring in 6 carbons into 2 3-carbon moieties. Okay, so because of the 2, that's why I specifically mentioned that there are 2 G3Ps, that's glyceraldehyde-3-phosphates per glucose. So we have 4. So how many bonds of ATP are produced? 4. And the net production therefore per glucose is 2, because we are utilizing 2 and we are creating 4. So eventually we get some energy after we break down a molecule of glucose. Okay, so this is what we have. We have 2 ATP expended, 4 ATP produced, that's 2 from each of the 2 3-carbon fragments from glucose, and the net production there is 2 phosphate bonds of ATP, or 2 ATP rather, per glucose. Okay, so our overall step is going to be glucose plus 2 NAD plus. Where did we use this NAD plus? We used 1 here. Is it just 1 here? 2 here, because we have again 2 of G3P. Okay, so this is where we are using 2 of them. Okay, then 2 ADP and 2 PI. Where was PI used again? In the same step where you had NAD plus, we used a PI to form the phospho. And what does this finally form? 2 pyruvate form 1 glucose, 2 3-carbon moieties, 2 NADH plus 2 ATP. So that is our total pathway. Now in aerobic organisms, you have pyruvate that is produced in glycolysis oxidized to carbon dioxide. That is what we are going to see when we do the Krebs cycle. This is also known as the TCA cycle. And the NADH, you have NADH produced here. This NADH that is produced in glycolysis and also in the Krebs cycle is reoxidized. What does it mean to be reoxidizing NADH? Means you have to get back NAD plus. Okay, because you need NAD plus for the glycolysis, and now it's already NADH. The only way you can get it back is via the respiratory chain. That's the oxidative phosphorylation which we studied with production of a lot of additional ATP, which we studied with the proton pump. Okay, there you saw there was mainly FADH and NADH that was used there, right? Now, another thing that we have to look at is anaerobic organisms. When we have aerobic organisms, we have the formation of pyruvate, okay? But in this case, what happens, there is the formation of lactate instead of pyruvate, okay? Where, for example, even in our muscles where there is a lack of oxygen at times, pyruvate is formed into lactate, okay? Now, in anaerobic organisms, what does anaerobic organism means? They function without the presence of oxygen, okay? They lack a respiratory chain in that sense, but in the formation of the glyceraldehyde-3-phosphate 2,1,3 bisphosphoglycerate, where we have the NAD plus and the PI, this is one of the reactions in glycolysis, okay? Now, the NADH produced in glycolysis must be reoxidized through some other reaction because it is needed for this reaction, and the NADH is reoxidized as pyruvate is converted to a more reduced compound later on that may be excreted. This is one of the cases, and the complete pathway including glycolysis and the reoxidation is actually called fermentation. We will see that. What we have here is now we created pyruvate from glucose. That was our final step in a glycolytic cycle, right? Now, we can reform NAD+. Remember, what did we take in the bisphosphoglycerate formation? We took NAD+ and made NADH out of it, right? Now, lactate dehydrogenases, it actually reduces the keto in pyruvate, and in the event it oxidizes NADH to NAD+. Again, it's a redox reaction, so the enzyme is going to be a dehydrogenase, right? Now, for example, this is what happens in skeletal muscles that ferment glucose to lactate during exercise when aerobic metabolism cannot keep up with the energy requirement. So, what happens is this pyruvate is further, what happens, there is a reduction of the keto in the pyruvate, okay, and this reduction will eventually do what? Oxidize NADH to NAD+, okay. So, the lactate released to the blood may be taken up by other tissues or by the muscle after exercise and converted via the reversible lactate dehydrogenase back to pyruvate to get into the Krebs cycle, okay. So, this is sort of a stopgap situation where we would have in the lack of aerobic metabolism that would lead to the lactate formation where there is not enough oxygen present. Now, some anaerobic organisms metabolize pyruvate to ethanol. This process you have learned, fermentation, like yeast. This is a reaction that yeast often do, where you have industrial chemistry where you learn about a lot of this, where we have NADH go to NAD+ by alcohol dehydrogenase. In the first step, the pyruvate loses carbon dioxide to form acetaldehyde, okay. That is one step of the reaction. This acetaldehyde then, in the presence of alcohol dehydrogenase, forms ethanol, okay, and in the event NADH is oxidized to NAD+. So, they metabolize pyruvate to ethanol, which is actually a waste product, okay. This is a process in fermentation. So, animal cells also contain alcohol dehydrogenase, which oxidizes ethanol to acetaldehyde, okay. So, we have ethanol, then we have NAD+, that is getting reduced to NADH, and what is it doing? It is oxidizing ethanol to acetaldehyde, okay, and then further oxidation is also possible to acetate, where you have NAD+ going to NADH plus H+. So, what are you doing in this case? You are reducing NAD+. In the event oxidizing acetaldehyde to acetate. In fact, the animal cells that contain alcohol dehydrogenase in the forming of acetaldehyde, this is what results in the hangover that you get from alcohol consumption. You hear of hangovers from alcohol consumption. What happens is this ethanol gets oxidized to acetaldehyde in the body by this particular enzyme alcohol dehydrogenase, and this acetaldehyde actually reacts with certain proteins to give you that numb feeling. Okay, so it's all biochemistry that goes on. So this is our final steps of glycolysis, where we have our glucose 2 ADP 2 NAD plus going to 2 phosphate plus 2 phosphate, that forms 2 pyruvate 2 ATP 2 NADH plus 2H plus. In the fermentation, instead of pyruvate we are going to get lactate. Okay, so we have the glucose that with 2 ADP plus 2 PI will give us 2 lactate plus 2 ATP, because remember, when we are forming the pyruvate to the lactate, if we just go back a step, this NADH NAD plus does not come into the picture, right? Because if we look at this reaction, we had the NAD plus and the PI here forming NADH, but that is not, the NADH is utilized in that pyruvate reaction to lactate. Okay, so we do not have the NAD plus NADH feature in the fermentation where we are creating lactate from glucose. Okay, because that pyruvate to lactate conversion requires NAD plus NADH. Okay. So if we now look at all the enzymes that were involved, there are 10 enzymes here for the 10 steps in glycolysis. Hexokinase, isomerase. So now you should be able to tell me exactly what each of these steps are doing. Hexokinase creates glucose 6-phosphate from glucose. Isomerase creates fructose 6-phosphate from glucose 6-phosphate. Phosphofructokinase creates the fructose 1,6-bisphosphate from the fructose 6-phosphate. Aldolase breaks it up into dihydroxyacetone phosphate and glyceraldehyde-3-phosphate. The isomerase interconverts dihydroxyacetone phosphate and glyceraldehyde-3-phosphate. Then we have glyceraldehyde-3-phosphate dehydrogenase. Then we have phosphoglycerate kinase that is going to create the bisphosphoglycerate. The bisphosphoglycerate is then has a mutase acting on it, where it creates from 3-phosphoglycerate it goes to 2-phosphoglycerate. Then we have the enolase that creates the phosphoenol pyruvate that then loses, because when you have the enolase you have to have an enol present there. So you have a phosphoenol pyruvate. That phosphoenol pyruvate loses the phosphorus by pyruvate kinase to form pyruvate. So these are all the steps of glycolysis. Okay, and if we now look at the specific free energy changes involved in the steps, we have a delta G0 prime. The prime means a biological standard where the temperature is taken as 37 degrees centigrade instead of 25 degrees centigrade, because you are looking at the free energy changes that are happening in the body when the reactions are taking place. In each of the kinase reactions, this is just the delta G with the concentrations that we have, this is delta G0. So how do we calculate delta G using delta G0? And you need the concentrations of the reactants and the products. That's all you need. How do? You calculate an equilibrium constant for a delta G0 process, right? And then you have the reaction quotient that is going to depend upon the concentration of the reactants and products that you have, and you can calculate a delta G associated with the process provided you know the delta G0. Fine. So considering that the concentrations of the products and the reactants can be calculated or can be obtained, we get a delta G associated with these enzymes, 10 of these enzymes. And if you notice, the kinase enzymes are the ones that are most spontaneous. Okay, they have high kilojoule per mole value, and the reason why they have this is because they have, this is a highly spontaneous reaction where it is going to lose the phosphate. These are the ones that use coupled reactions with the ATP that provides the energy actually to give you the product. Okay. So what we have is, if we look now at the energetics of phosphorylation, we are looking at the first step, hexokinase. Okay. The glucose actually needs the phosphate to become glucose 6-phosphate. The delta G0 associated with that is actually plus 14 kilojoules per mole. So there is no way it's going to happen by itself. Okay. But you have a coupled reaction, that is ATP, the hydrolysis of ATP, the high energy phosphate bond, that is actually going to break ATP into ADP plus PI. This PI is going to be supplied to glucose. This free energy change is minus 30 kilojoules per mole. Okay. So we have this coupled reaction. So we use the half reactions to determine the delta G zeros for the whole reactions. Okay. And we have therefore an overall reaction that is going to be glucose plus ATP, giving us glucose 6-phosphate plus ADP with a favorable free energy, standard free energy change that we have here. Okay. Now, depending on the concentrations of glucose 6-phosphate that you have and the concentrations of glucose and the relative amounts of ATP and ADP that you have, the free energy change of the reaction is going to change, right? Because what is that reaction that we have? If we look at a delta G0 value, now what you can have is, for each of these you can have an E0 also associated with it, because remember you have a redox reaction taking place. Okay. In some cases you would have a redox reaction taking place where you would have this minus NFE, right? From this you can determine the equilibrium constant. What is the reaction equation? Equilibrium, right? Then you can have a delta G. What is that? That is delta G0 plus RT ln Q. What is this Q? This Q is your reaction quotient. What is the reaction quotient? It is the ratio of the concentrations of the products to the reactants. Okay. So based on that you can find the delta G associated with this. Now if we look at this first reaction that we have here, the fact that glucose 6-phosphate concentration is low, okay, what is that Q? In this case, what is the Q going to be? It is going to be glucose 6-phosphate ADP divided by glucose ATP concentrations. That is what Q is going to be. Now if you have a high concentration of products, that could more than compensate the delta G0 value negative that you have here and make an overall delta G of the reaction positive. Okay. What am I talking about? I am talking about this factor. This is negative, but the ratio of the products to the reactants is important here. Okay. You cannot have too higher concentration of the products, because that will overcompensate for the negative delta G0 that you have here, making this non-spontaneous. Okay. This is what happens in most biological reactions. The product concentrations are pretty low, making the reaction spontaneous because your delta G0 of the coupled reaction is negative. Okay. So the concentration of glucose 6-phosphate in this particular reaction is actually low, and the overall Gibbs free energy that you get is minus 27 kilojoules per mole. So what you can do with this value is actually determine what the ratio of the products to the reactants are. You know what delta G0 is, you know what delta G is, right? You can find the ratio of the products to the reactants, and when you are considering, if it is not mentioned, a delta G0 prime value means you use a temperature of 37 degrees centigrade. Okay. So, that would be how you would actually calculate the energetics of every step of glycolysis. Okay. We can calculate the energetics associated with every step of glycolysis and find an overall energetics associated with this. Now, the spontaneous reactions that we did see, that was with this particular case, this enzyme is hexokinase, right, and the other ones that I showed you were phosphofructokinase and pyruvate kinase. Okay. These are the three enzymes that actually catalyze spontaneous reactions, and the control of these enzymes actually determines the rate of glycolysis. Okay, because each of these particular reactions, if you go back and look at the glycolysis pathway, it is these three reactions that are irreversible. So, once glucose forms glucose 6-phosphate, it is trapped in the cell. Okay. So, the control of these enzymes actually determines the rate of the glycolysis pathway. So, there are two types of control. We have local control and global control. Local control means that it involves the dependence of the enzyme catalyzed reactions on concentrations of the pathway substrates or intermediates within a cell. So, what is this local control? This is exactly what I was speaking about in the last slide, where we have the delta G values associated with the amount of product and reactant that you have. Okay. So, if my product concentration gets too high, what is going to happen? My reaction will be non-spontaneous. Okay. So, that is where you would have local control. If glucose 6-phosphate has too high a concentration, the delta G associated with the expression that we have written here will make this non-spontaneous. Okay. So, this is regulated by the amount of glucose 6-phosphate that is formed, because remember I mentioned in the last class that glucose 6-phosphate actually inhibits hexokinase. So, what is going to happen? If it inhibits hexokinase, then this enzyme cannot act on another glucose molecule, which means that the concentration of glucose 6-phosphate is getting too high, making this Q too large, which is offsetting the delta G0, preventing a reaction from taking place. Okay. You do not want that to happen. Okay. If you want the glycolysis reaction to go forward, then you have to have this regulation. You do not want too much of glucose to be broken down. Okay. That is where the regulation takes place. Okay. So, we have local control, which involves the dependence of enzyme catalyzed reactions on concentrations of pathway substrates or intermediates within a cell. You now realize what this means, that the concentrations of the intermediates, the concentrations of the products and the substrates are extremely important in determining the dependence of these catalyzed reactions. Then you have global control that actually involves hormone activated production of second messengers. These are certain messengers that are there in the body that actually regulate the cellular reactions for the benefit of the organism as a whole. So, this is like a more global approach where it would work on the cell itself, preventing the cell from acting or preventing certain reactions taking place altogether. Okay. But at the local level, the concentration dependencies are due to the variations in the product and the reactant. Okay. Now, let us look at one such example. This is one step of the glycolytic pathway. Okay. If you look at this, you see it has two phosphates attached, carbon number 1 and carbon number 6. You have a ketose. So, this is fructose 1,6-bisphosphate. It has broken down into dihydroxyacetone phosphate and glyceraldehyde-3-phosphate. So, the enzyme that has acted here is aldolase, okay, in a reverse aldol reaction. So this is what you should be able to recognize. This is the key step in the glycolysis. Why? Because it is actually breaking down the glucose. Previously, all the steps that led to fructose 1,6-bisphosphate still had the 6 carbon atoms in them. Okay? This step, the aldolase step, is the one that you are breaking the 6 carbon unit into 3 carbon units. Okay. Now, we have a delta G0 of this particular reaction having a value of plus 23.8 kilojoules per mole. Okay. The Gibbs free energy is actually favorable. What does that mean? I have a delta G, let's just write this down clearly. So I have a delta G and I have a delta G0 plus RT ln Q. This value is plus 23.8 kilojoules per mole. This value is minus 6 kilojoules per mole. So where is the regulation coming from? It has to come from Q, right? So what we can do is we can actually calculate, assuming that fructose 1,6-bisphosphate is 1 millimolar. I have these in equal concentration, right? Because it's breaking down, it's forming from this. So I can actually find out the concentrations of glyceraldehyde-3-phosphate and dihydroxyacetone phosphate at equilibrium. How can I do that at equilibrium? It will just be the equilibrium constant that I get from the delta G0 value. What do I get from the delta G0 value? I forgot to write a minus in the first one. I don't know if you pointed it out. Okay. So I can get this because I know what my delta G0 is. I can also get, so I can get the ratio of the products to the reactants based on considering a 1 millimolar concentration of fructose 1,6-bisphosphate, and I can also find the same ratio during glycolysis where my reaction is actually favorable. Okay. So this whole idea gives you the energetics that are associated with each of these steps. Each of these steps has associated with it this energy. Okay. So each of these energy values can tell me what is actually happening and what delta G0 the reaction actually had, the equilibrium. Okay. So this completes our discussion on glycolysis, where we have actually broken down glucose to form pyruvate. Okay. Our next step will now be to see how there is further degradation of pyruvate in the tricarboxylic acid cycle, where finally carbon dioxide and water will be formed. We will do that in our next class. Thank you. In the last step of our metabolism of carbohydrates, we are going to consider today the tricarboxylic acid cycle or the Krebs cycle. Now, if we look at the 3 stages of cellular respiration, there are actually the first stage that we have gone through, that is glycolysis for acetyl coenzyme A production. We will see, we have already seen the breakdown of glucose to pyruvate, and we will see now how pyruvate actually gets to acetyl coenzyme A, that then gets into the tricarboxylic acid cycle, which eventually leads to the production of carbon dioxide. And we have already considered the third step, where we have electron transport and oxidative phosphorylation that leads to the production of ATP. So these are actually the 3 stages of cellular respiration, and if we consider the catabolic pathways of all the breakdown, whether it's amino acids or fatty acids or glucose, they ultimately lead to acetyl-CoA, which is the key component of the TCA cycle, which we will see. So what happens is, what we have studied in glycolysis is glucose getting to pyruvate. Now the reverse of this is also possible in a process that is called gluconeogenesis, where glucose is formed from pyruvate. It is not the exact reverse of the glycolysis steps, but relatively, since these involved some of the similar enzymes, we do have the pyruvate getting back to the glucose. So we also saw how the lactate is formed from the pyruvate. So we have lactate also getting to pyruvate, and amino acids have actually entry points at 3 points, where it can get to pyruvate, you can get to acetyl-CoA, or you could get directly to the tricarboxylic acid cycle. Fatty acids get into acetyl-CoA. Okay? So we have considered so far the breakdown of glucose to pyruvate. Now in general, we have these 3 overall steps. All of them produce ATP. Okay, that is our major concern in the production of energy. So this is the lysine. So we see that, you see the lysine here, and what are these chains? These are the lipoic acid chains. Can you see the lipoic acid chains? These are the lipoic acid chains that have the dithiol, and the dithiol, one of them picks up the acetyl and has it linked to the sulfur of the lipoamide, right? The lipoic acid part here. Now what happens is, coenzyme A now comes into the picture. Coenzyme A now has to form acetyl-CoA, so it picks up this acetyl, forming acetyl-CoA, and then you have the reduced SH. Now what must happen for this to act again? This has to get back to the dithiol there. So two of these H's have to be removed. How can they be removed? They are removed by FAD. FAD then comes into the picture, picks up the 2 hydrogens, and this gets back to your oxidized lipoyl lysine. Is that clear? So this is just a generic procedure where you would have these to, because you realize that you have the acetate formed here directly already, but you have to have these enzymatic steps to get the enzymes back to where they started from, so that they can go and act on another pyruvate. Eventually what happens is, this gets to the oxidized part, so this can now take up another acyl rather. Okay, so another acyl can now get attached to this. FAD has now been reduced to FADH2. What